cross-section through one skeletal muscle cell right here. So, what's this?
Epimycin?
Pardon?
Epi?
No, this is one cell.
Oh.
This is a single cell. So what's the cell membrane called? Sarcolemma. Sarcolemma. So this is the cell membrane. This is the sarcolemma. And then what is the innermost layer of connective tissue that wraps around the cell? Innermost. 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 Endomysium. Endomysium connective tissue. That's what this is. Endomysium connective tissue. This view here, this would be the end of an axon collateral from a somatic motor neuron. Right here, you can see that they are all myelinated. They all have myelinated axons, Schwann cell formed myelin sheaths. So talodendron leading to an axon terminal right there. Talodendron, axon terminal. So inside here, will be a bunch of synaptic vesicles full of ACH. Now the motor end plate, the motor end plate is a portion of the sarcolemma. It's a region of the sarcolemma. What this model doesn't show you, what that model doesn't show you is the motor end plate is very folded. The portion of the sarcolemma actually ripples. It actually forms many, many folds. That increases the total surface area for nicotinic cholinergic receptors. The more folds, the more receptors you can pack into that portion of the sarcolemma. So, see the folds right there, folds here, and so on. So all the folded portion of the sarcolemma collectively would represent the motor end plate. That's also where all the nicotinic cholinergic receptors are located. So action potential, subthalamic conduction, node of Ranvier, the telodendria, the net movement of calcium in, exocytosis of acetylcholine, all binding to nicotinic cholinergic receptors, gates open, sodium in, depolarization beyond, so depolarizing low potential spreads out, sarcolemma here and here. Where the voltage gated channels are, they open up, action potential spreading in both directions along the sarcolemma away from the motor end plate. Okay, what else we say here? We can see shallow nucleus, it's the shallow nucleus, multiple shallow nuclei. And on view, cross-sectional view, every brown dot represents a myofibril, one myofibril. And what they get wrong is something that this model gets right. You can see how the myofibrils are packed right up against each other. You don't see any obvious room in between them. That's accurate. The interior, you also have mitochondria wedged in there. You've got an organelle called the sarcoplasmic reticulum wedged in there. But the myofibrils take up virtually all of the internal volume of the cell. So they should be, so not only should they be packed pretty much against each other, these gaps in between are fantasy. These gaps do not exist. So I can understand the model maker not knowing microanatomy, making that mistake, but whoever said, okay, it looks good to me, should have been fired. So again, every dot represents a cross-sectional view through one myofibril. Every dot is that cross-sectional view through one myofibril. So the longitudinal view reveals maybe 20 or so. I'm not going to count them. It looks like about 20 myofibrils. Again, running the entire internal length of the skeletal myofiber or cell. And you can see the alternating A and I bands. There's an I band right there with a Z disc. There's an A band with an M line. I band, A band, I band, A band, I band, A band, I band, A band. And the sarcomere is one portion of a myofibril from Z to Z. So from the middle of an eye to the middle of an eye. So that's one complete sarcomere. That's one complete contractile subunit. So this could contract, that could contract, that could contract, and so on. Excuse me. Let's see. Now what they get wrong here Another thing that this model gets wrong, one thing this model gets wrong, is that the Z disc is a huge protein sheet. It's an enormous protein sheet. Here's a Z disc, and it's continuous. That Z continues to this. It's one thing. This would be one complete Z disc going through the entire cell, attaching to the membrane here, attaching to the membrane here, and going all the way around, attaching to the membrane all the way around. So that sheet goes through the whole cell. The next Z disc does the exact same thing. The next Z disc does that as well, and so on. So they're large. Each Z disc is one really large protein sheet. The M lines, on the other hand, the center of an A band, is a tiny protein sheet that only runs through an A band. So that's one M line, that's a separate M line, that's a separate M line, and so on. So an M line does not extend from A to A to A, the way a Z disc extends from I to I to I. So it's a much smaller protein sheet, just holding the A band together. Okay, anything else on here? I think that's about it for this model.
So
Questions?
there would be myofilaments in the...
Yeah, that's exactly right. The myofibril would be composed of many, many thick and thin myofilaments.
Okay.
All right, as we continue going to increasing magnification, they're going to become apparent. Okay, not quite yet. We're not quite magnified enough yet.